Prostate cancer occurs as a result of an accumulation of mutations within the DNA of a prostate acinar cell, which allows the cells to proliferate uncontrollably. Hanahan and Weinberg describe several hallmarks which cancer cells acquire through these mutations in order to survive, proliferate, and sometimes migrate. Two such important hallmarks are angiogenesis and sustaining of proliferative signals. In normal tissue, growth signals are tightly regulated to prevent abnormal growth, and hence cancer cells require mutations that allow them to maintain or enhance proliferative signals. This is achieved through mutations which affect the proliferative signaling pathways that are initiated by growth factors or direct contact with extracellular matrix components. Angiogenesis is required to support the growing tumor mass that demands increased nutrients and waste clearance for chronically proliferating cells. These cells expand existing vasculature by switching on pro-angiogenic factors to induce normally quiescent vasculature to initiate endothelial cell proliferation, capillary sprouting, and vessel branching. The sigma-1 receptor is a ubiquitously expressed chaperone protein, usually inert in normal conditions. However, a unique phenotype is expressed and active in prostate cancer cells. The active protein plays an important role in influencing nearly all of the hallmarks associated with cancer, including sustained proliferative signaling and induction of angiogenesis, and hence represents a promising target for anti-cancer therapy. Activation of the sigma receptor has an amplifying effect within the cell, which increases the transduction of calcium and its concentration. Endogenous activation of the sigma receptor activates a downstream chaperone protein, which results in a cascade of events increasing cytosolic concentrations of phospholipase C, an enzyme which mediates the hydrolysis of the substrate PIP2 to diacylglycerol and IP3. Diacylglycerol activates protein kinase C, whilst an increase in cytosolic concentrations of IP3 causes enhanced binding to IP3 receptor-mediated calcium channels on the endoplasmic reticulum, which facilitates the movement of calcium from the endoplasmic reticulum to the cytosol, activating protein kinase C. Protein kinase C binds to and activates importin beta. When this complex becomes active, it can transport cytoplasmic HIF-1 alpha through a nuclear pore into the nucleus, whereby it binds to specific sequences in the genome to allow expression of pro-angiogenic factors. This pathway is heightened in carcinomas pertaining to the prostate and thus increases angiogenesis.
The sigma-1 receptor is also involved in the process of transducing important signals, including those required for proliferation from the extracellular matrix through beta integrin. One method by which this is achieved is through the action of sigma-1 as a chaperone for the potassium channel hERG. Sigma-1 translocates hERG to the cell membrane and facilitates the formation of a complex between hERG and beta integrin. This beta integrin hERG complex uses two main methods in order to transduce signals induced by binding of beta integrin with extracellular matrix components. One of these methods involves the formation of large multi-protein complexes with various signaling molecules that can activate downstream pathways that influence angiogenesis, proliferation, cell growth, survival, and motility. The other main pathway is especially important in maintaining cell growth signals. In this pathway, when beta integrin bind extracellular matrix, it activates hERG, allowing potassium ions to move down their concentration gradient out of the cell, thus causing hyperpolarization. This hyperpolarized cell environment favors the movement of cations such as calcium and sodium down their electrochemical gradient. Calcium influx stimulates the transcription of mitogenic factors which stimulate the progression of the cell to rest phase, thus inducing cell proliferation. Sodium influx is important for supplying nutrients and substrates required to support progression through the cell cycle, as these often travel into the cell with the ion. Thus, through enabling the formation of beta integrin hERG complexes, the sigma-1 receptor initiates and supports the proliferation of prostate cancer cells. A small molecule inhibitor that binds the unique sigma receptor phenotype will be developed to selectively block the action of the protein within prostate cancer cells. Inhibition of the sigma-1 receptor will decrease subsequent HIF-1 alpha translocation to the nucleus, thereby decreasing the production of pro-angiogenic factors. As a result, the target cell will be isolated from the blood supply and unable to obtain sufficient oxygen and nutrients to support cell growth and proliferation. Inhibition of the sigma receptor will simultaneously reduce the proliferative, angiogenic, survival, and migration signals provided by direct contact with extracellular matrix components via the hERG potassium complex pathway. Some possible limitations to our approach is that the cancer cells rely on multiple pathways to achieve the same results, and thus blocking the pathways involved in the sigma receptor may not be sufficient. Furthermore, the multi-protein complexes formed by the initial sigma-1 mediated hERG beta integrin complex are numerous, and only some of these are well studied and understood in a small selection of cancer phenotypes. Thus, these pathways could have anti-tumorigenic components, whereby inhibition would lead to increased tumorigenesis if the cancer cells were to survive.